Good morning, everyone. I'm Liz Fernandez with the Professional Development Services Branch, and I'd also like to welcome you to today's webinar. So we have 3 speakers for our webinar today. Dr. Tom Gidlewski is a veterinary pathologist and the lead of the Wildlife Services National Wildlife Disease Program with APHIS in Fort Collins, Colorado. Dr. Julie Lenoch is a veterinary epidemiologist and a member of the Field Epidemiology Investigation Services with APHIS VF, also in Fort Collins. And Dr. Lynn Creekmore is a veterinary epidemiologist for aquaculture and non-program species within aquaculture, swine, equine, and the poultry center with APHIS VF in Fort Collins, Colorado. And with that, I'm going to turn the webinar over to Dr. Julie Lenoch.
Thanks, Liz. Quick audio check. Are we good? All right. Thank you, and hopefully you're in the right webinar. You're here to learn about rabbit hemorrhagic disease virus 2. I'll be kicking us off, but we're going to do this as a combined presentation. So a roadmap of what we're going to do today, I'm going to start and give an overview of RHDV2 and then discuss our formation of our coordinating cell and how we've been attacking this problem, and then review what we were given kind of as our marching orders for the goals and responsibilities for the team. I'll then hand it off to Tom, and he will give a good amount of information on the wildlife experimental studies that have been done, and then the wildlife combined response for detection and response. Lynn will go into details on the Southwest outbreak and then give further details in the weeds on the epi and vaccine use, and then I'll kind of wrap us up, and we'll all be available for question and answer. I'm getting a note in the chat that they can't hear anything. Are we good?
Julie, I will instruct them to, ladies and gentlemen, please be sure to dial in for the audio as well. I will provide those instructions in the chat right now.
Thank you. Okay. Quick overview. So rabbit hemorrhagic disease is a rabbit colici virus. There's 2 recognized strains. The first one that was discovered is now called RHDV-A, previously called RHDV1. And for today's talk, we're talking about RHDV2, which has really been known for about the last 10 years. About 2010 was the detection for the second strain, and that's the focus of our talk today. RHDV2 is fatal to domestic, feral domestic, and multiple wildlife species. And again, Dr. Gidlewski will give more details on that part of it. This is a pretty significantly environmentally hardy virus. Some studies show that it can survive in the environment for 90 days or more, and that certainly poses some problems with biosecurity and some disease mitigation. And we're currently looking at it being in the United States for about the last 2.5 years. We had isolated events in 2018, 19, and 20 in Ohio, Washington State, and New York City. And then we have an ongoing and geographically spread outbreak in the 7 southwestern states, New Mexico, Arizona, Texas, Colorado, California, Nevada, and Utah. And Saqib, if you can go to the video, now would be a good time. So our GIS team within CIA, the Center for Epidemiology and Animal Health, created this video for us on our case detections over time. And we'll pull that up and switch over. Perfect. Okay, so the blue dots in the upper left corner are actually in Canada. Those are in British Columbia, popping up very, very close to the Washington State border. And then again, we had a single premise in Medina County, Ohio. Focus your attention back on the Pacific Northwest, as you'll see some dots pop up again in Canada, and then in the San Juan and Seattle area through 2019. We're pretty isolated up in that region. Then New York pops up, and our Southwest disease spread. and that was about six months there. Perfect. So this is just a screenshot of that final slide. So I think everyone can see the problem that we're dealing with, that those Southwest states, we're looking at close to 150 separate detections in the 7 Southwest states. These are American detections, and Lynn will give a little bit more information, but we're seeing similar disease south of the border in the northern states of Mexico. Okay, so what's at risk? This was a bit complicated at the beginning, in part because we just didn't have a great grasp on what our commodity groups looked like and kind of how diverse the stakeholders are. So because we have both domestic and wild species involved, understanding where the domestic rabbits are and kind of numbers. So according to the AVMA, there's probably around 6 million pet rabbits in the United States. Our commodity groups, which are typically what's thought of more as our slice of the pie as far as the agricultural part, we have animals raised in the U.S. for meat, and that is both for human consumption, rabbit meat, and for the pet food market. Fur is a declining market, but still present in the U.S., so that's a terminal activity. The rabbits are raised and then skinned for rabbit fur coats and that type of thing. And again, that's been going down in popularity for about the last 20 years. Rabbits are also raised for wool. Angora rabbits are harvested for their fiber, and that market has been growing over the last 15, 20 years. And then we have a multitude of rabbits that are raised for biologics, laboratory use, industry type of things. So I kind of bunched all those under biologics. So those are more of what we would consider typically our agricultural rabbits. There's also about 900,000 show rabbits in the U.S. We have rabbits raised for breeding, and the American Rabbit Breeders Association recognizes 49 different breeds of rabbits, and the United States is thought of as the primary feedstock for many of those breeds. And we've been discovering that we have a pretty large and integrated rabbit rescue network, really through North America, but that can involve some multiple states and some movements of rabbits on the rescue side. And then on the wild lagomorphs, we have the rabbits, cottontails, and jackrabbits, or our hares, and then pica also fall under the wild lagomorph category. We have not yet seen detection in pica, but believe that there may be susceptibility to RHDV2. So the reason that you're listening to this and the reason that we're talking about it is because we can, because it's a bit complicated. We have a transboundary disease right now. Again, we've had detections in Canada, the United States, and in Mexico. We know that we have involvement right now of both wild and feral rabbits, and that makes disease mitigation, detection, and proving freedom from disease quite a bit more complicated. A big picture issue is that we have an OIE reporting responsibility, and simultaneously we lack regulatory authority over rabbits at the national level, and the states have a little bit of a mixed bag as far as regulatory authority. The diverse rabbit sector that I just talked about presents some unique challenges for how to get messages out to them, how to share our information via social media and other options to get information out to the right folks. And then Lynn will go a bit more into the challenges that we faced with vaccine and laboratory testing. So for those reasons, at the very beginning of this, I'll go back to maybe March or April of 2020, APHIS formed a coordinating cell, and the team has had involvement from all 3 business units from Veterinary Services, that's strategy and policy, field operations, and the diagnostics and biologics team. And very quickly we realized that Wildlife Services needed to be at the table, and we were delighted that Tom Gidlewski has stepped up and been our point of contact there. So this has truly been a combined effort between Veterinary Services and Wildlife Services from the very beginning. just kind of a general visual of what we were trying to do to improve disease awareness and prevention. We worked on some outreach biosecurity and really have been working with our international partners, again, both Canada and Mexico here, but truly Australia, New Zealand, Europe, and some other folks as well. Lynn will go into more details on the epi investigations and a summary of what our diagnostics and biologics teams have been doing. And then again, Tom will kind of get a little bit more in the weeds on the wild rabbit activities. So with that, I'm going to pass the torch to Wildlife Services. And Tom Gidlewski, are you up and ready?
You betcha. Thank you, Julia. And as Julia mentioned, we really do appreciate the opportunity to be with you here today. Next slide, please. We've demonstrated this disease at Plum for a long time, and it's fairly characteristic in that we can often go in with the students to examine the rack of rabbits, and when we walk into the room, every rabbit looks good. And before we're done our 5-minute examination of these rabbits, we'll often have an animal throw himself to the side, begin to paddle, vocalize, and the animal is dead before we can ever draw up euthanasia solution. So this disease is often acute or peracute. These animals are in good body condition. And in addition to the features we described, we'll often see this foamy, bloody nasal discharge coming out the nose. Next, please. So peracute is usually a condition in which they're encountered, usually found in good body condition after death. Most severe lesions of hemorrhagic disease are found in the liver, trachea, and lungs, and petechia or hemorrhages are evident in almost all organs, and are the result of disseminated intravascular coagulation with its associated poor blood coagulation. Next, please. The main route of infection is oral, followed by the conjunctival and respiratory routes. Liver, spleen, and lung are the primary target organs, and liver has the highest titer of the virus. Next, please. Here we have some really nice lesions. Up on the upper right, you'll see a slide that contains a mahogany-colored normal liver, and next to it is the pale brown liver. And when you go to the far left, you can see a close-up on that liver, and you can appreciate that paleness and also that reticular or interstitial pattern. Below that slide, so down there in the lower left, is even a further close-up, and you can really appreciate that acinar or reticular pattern of hepatic necrosis. Very, very severe hepatic necrosis. Sometimes it's hard to appreciate because you can't see a line of delineation between the normal and the affected. In most cases, those livers are totally hepatically necrotic and totally involved. On the lower right, we have a similar pale yellow liver, but in this case, we also have mineralization going on as well. Next slide, please. Just some really nice lesions, and I apologize for these lesions, but it's in my contract. I always have to show lesions. So we've got some heart lesions on the epicardial surface. We have some lesions, some hemorrhages. We have hemorrhages on the serosal or outer surface of the bowel. Once again, that upper right-hand slide demonstrates the difference between the normal and the abnormal liver. And then we see hemorrhages on the lung, as well as hemorrhages in the trachea. And we also have an area on that kidney down there where we have an infarct because of the thrombosis. So we have infarct, and we have an area that is now devoid of blood and necrotic. Next slide, please. So when it comes to differential diagnosis on these cases, we want to be really careful to think about the zoonotic diseases such as tularemia and plague, make sure we're using the appropriate PPE. But one point that I really want to make about this disease dovetails back with what Julia said. The agent is very, very hardy, and we want to be really careful to use appropriate biosecurity when we're handling these carcasses because they're full of the agent, and that agent persists for a very long time. Like Julia was saying, up to 90 days. So we want to be very careful that they're handled appropriately, don't find their way back out into the wild to re-disseminate the disease. Next slide, please. So as Julia mentioned, rabbit hemorrhagic disease 1, or A, has been recognized since about the mid-'80s. It quickly spread throughout Europe, and it was soon determined that it was strictly a disease of the European rabbit. Some original research was done which demonstrated that our domestic rabbits and jacks were not susceptible to this virus. So rabbit hemorrhagic disease 1, or A, ran throughout the world from about the mid-'80s up until 2010. And then we started to see something going on. Vaccinated rabbits were breaking with disease, and younger rabbits were coming down with the disease. So it appeared that something had changed. And sure enough, it had. Rabbit hemorrhagic disease 1 was still circulating, but there was a new variety, rabbit hemorrhagic disease 2. Once again, that was 2010, and this disease readily spread throughout the world as well, just like one did originally. However, the host range was broader with this disease, and it attacked younger rabbits. So in 2017, in one of our FAD schools, I got to visiting with Fazee Mohammed, the pathologist out there at Plum, and we decided that we should probably redo that original research and find out if our wild rabbits are indeed resistant, or maybe they might be susceptible to this new variant, this rabbit hemorrhagic disease 2. So we got Jeff Root, a small mammal researcher, disease researcher here at the National Wildlife Research Center, and we went out to Long Island and we collected quite a few eastern cottontails. We took those eastern cottontails over to Plum and inoculated them with rabbit hemorrhagic disease 2, and lo and behold, they were indeed susceptible. We had about a 60% attack rate. The lesions were very similar to what we saw with rabbit hemorrhagic disease 1 and rabbit hemorrhagic disease 2 in European rabbits. The only difference that we noted was that the course was a little bit longer. It took an extra day to develop the lesions and mortalities. Next slide, please. So some of the lessons that we learned from Europe was that there are caliciviruses that normally circulate throughout the rabbit populations. And depending on whether or not these rabbits have seen these other caliciviruses can affect the outcome of their exposure to rabbit hemorrhagic disease. They may have some immunity from having experienced these less lethal caliciviruses in the past. Another thing that they noticed in Europe is that when the disease gets entrenched within the wild rabbits, it becomes a lot tougher to eliminate, and in many cases, they have to live with the disease as compared to the situation when the disease is limited to domestic rabbits. Next slide, please. So that was 2017. We did that research, and we kind of thought nothing. We thought maybe we dodged a bullet. It had been 3 years, and we hadn't seen any infections in our in our wild rabbits. But then, lo and behold, in March of 2020, that came to an end, and we started to see cases of mortality in our wild rabbits. Here's an example in our antelope jackrabbit, our mountain cottontails as well. Next slide, please. Our black-footed jackrabbit and our desert cottontail. And we also knew that our eastern cottontails were susceptible because of the research that we had done originally. So now we had a situation where we had to track that disease in wildlife so that APHIS could use that information for OIE reporting, as has been mentioned, as well as risk analysis for our domestic rabbits. So Wildlife Services has been tracking that disease in wildlife for APHIS, and I need to recognize Tim Linder and Jordan Ringenberg. They have been looking after the tracking of this disease, and I have to appreciate that they graciously tolerate my interference. So at any rate, that's been our role, is to try to work with the state wildlife agencies to keep track of this disease. The state wildlife agencies have been really great to work with, including their National Wildlife Agency. And in addition to that, we had a little meeting about a week or 10 days ago now where we visited with all the wildlife agencies that had seen mortalities. And it sounds like they're starting to see a lot less of a situation in their wild rabbits and hares. And it's too early to tell, but they kind of have the impression that the populations were not severely impacted. And luckily, we know from Australia and other places that have tried to use biologic control in their rabbits, that our rabbits and jacks are pretty resourceful, and hopefully they can bounce back from this disease. Okay, I think that was everything that I wanted to mention, Julia. Thank you.
Excellent. Thanks, Tom. So moving on from the summary from Wildlife Services, we're heading over to Dr. Lynn Creekmore, representing all of our folks in strategy and policy. Lynn, are you ready?
I'm ready. Thank you, Julie, and hello, everyone. I really appreciate the opportunity to speak with you today, and so let's dive into what happened this year. So on March 24th of this year, our NBSL, Foreign Animal Disease Diagnostic Laboratory in Plum Island, detected RHDV2 in tissue submitted from domestic rabbits from a premises in Cibola County, New Mexico. Earlier in the year and concurrent to the initial positive finding, mortality was also being reported in wild rabbits and hares in both New Mexico and Texas. Causes of mortality weren't determined for these early events, although tularemia and plague were ruled out for many of these wild lagomorph cases. Following that finding in the New Mexico domestic rabbits, and with RHDV2 added to everyone's differential list, RHDV2 was detected in samples submitted from a wild black-tailed jackrabbit from Eddy County, New Mexico, and from wild desert cottontail rabbits from Doña Ana County, New Mexico. These represented the first detections of RHDV2 in wild rabbits and hares in the U.S. Finally, at the same time, word began to come to us via our Mexican colleagues that mortality was also occurring in domestic and wild lagomorphs in Mexico. Next slide, please. So additional positive findings really quickly followed these initial findings in New Mexico. And at present, we have 7 states involved, and in alphabetical order, they're Arizona, California, Colorado, New Mexico, Nevada, Texas, and Utah. To orient you to this county-level map that's depicted, the blue shading that you see indicates the 45 counties with domestic rabbit detections, and where you see the hash marks or diagonal lines, that indicates 49 counties with wild rabbit detections. And where you can see the red outlines around counties, there's one in Utah and one in Colorado. Those are the 2 counties with feral rabbit detections. As you can see, for many of the counties, there have been detections in both wild and domestic rabbits. And we're in between sit reps, so if you see a discrepancy on this map where you think something's missing, that's probably why. We can't quite keep up real time with all that's going on. So as you can see, as I mentioned, both domestic and wild lagomorphs are involved in all of the states, and then in feral rabbits in 2 states, Colorado and Utah. Currently, there are 100 positive domestic rabbit premises and 78 wild rabbit detections across these 7 states, and again, those 2 feral rabbit detections in just the 2 counties in Colorado and Utah. The largest number of domestic rabbit detections, about 75% of them, are in Arizona and New Mexico. And the bulk of the wild rabbit detections have been in Arizona and Texas, followed by Colorado, California, and New Mexico. In Mexico, there are 7 states, Mexican states involved in the northern half of the country. And for those of you that are familiar with the Mexican states, the ones that have had detections in wild and domestic rabbits are Chihuahua, Sonora, Durango, Coahuila, Baja California Sur, and Aguascalientes. Next slide, Julie. So just a few comments on EPI. The source of disease introduction and the introduction point aren't known. I'm going to talk a little bit more about this when we get to the gene sequencing slide in a minute, and the gene sequencing work that's underway. Typical domestic rabbit husbandry practices are likely a contributor to the spread of the disease that we're seeing between domestic and wild populations both ways. as you can imagine, these states that are involved have warmer climates. So outside hutches and pens for the domestic rabbits are quite common. Some premises even have enclosures with burrows where the rabbits just live outside in sort of a natural environment. In addition, owners that have animals that are mostly housed inside will allow their rabbits to have some level of outside time as well. So that's another way that the populations can cross paths. The presence of the disease in wild rabbits definitely appears to be a significant risk factor for some premises, but we're finding that even rabbits that are strictly housed indoors on some premises that have been affected. So we really think there are some other risk pathways that may be in play, like mechanical vectors, fomites, maybe even forage or feed, for instance, hay that's grown in an affected area. There is a large amount that we still don't know about the epi of this disease. Our epidemiologists in the affected states, both on the state and federal side, have worked together to develop an information worksheet that's being deployed to collect key epi information, and hopefully that will get at some of the mysteries that we still have. We plan to analyze the results from that effort at some point and hope to get at risk factors, patterns, and maybe answer some of our more puzzling questions. So we look forward to being able to provide those findings to you in a future presentation. Next slide, please. Thank you. So I mentioned gene sequencing. Gene sequencing of RHDV2 isolates from Arizona, Colorado, New Mexico, and Texas has been completed by NVSL FADL. And in that work, they showed that these isolates form a single genetic cluster, suggesting that the outbreak of RHD in these states was caused by the introduction of a single genetic isolate into the region. Sequencing also showed that the same virus is responsible for the disease in both domestic rabbits and wild lagomorphs, so we're dealing with the same virus in both of those populations. Further, from looking at the isolates from those 4 states, they determined that the virus circulating in the Southwest is distinct from the RHDV2 isolates collected from domestic rabbits in New York, Washington State, and Ohio, as well as British Columbia, Canada. So the virus that in our big outbreak, our multi-state outbreak, is distinct from those that we saw in the northern states. Mexico's also identified their virus as RHDV2, and they've shared their findings by posting sequences to GeneBank. And finally, NVSL FADL is continuing our genetic sequencing work to compare additional U.S. isolates and to compare our isolates to others that are being found around the world. Next slide, please. There is no licensed RHDV vaccine in the U.S., but there are 2 killed vaccines licensed in the EU and used pretty widely in those countries. For these 2 vaccines, CVB is allowing importation into the U.S. with special permit for emergency use under the direction of the SAHOs in the states with confirmed cases. So far, the vaccine has been imported in all of the affected states. We are certainly hopeful that at some point we'll have a licensed vaccine for use in the U.S., and our Center for Veterinary Biologics is eager to work with companies that might be interested in U.S. licensure. This would be so much better for us in terms of our knowledge of safety and efficacy, and possibly also would be helpful in terms of cost to the rabbit owners utilizing the vaccine. We have a vaccine FAQ document that's available in our outreach materials, and Julie's going to tell you more about that in a little bit. Next slide, please. In terms of OIE reporting, we did report the initial detections in this outbreak to OIE way back in March, and we ended up attaching the report for this outbreak as a follow-on to the Washington and New York outbreaks. As you saw on the map that Julie showed earlier, in 2019, we sort of had follow-ons from Washington to New York to New Mexico in terms of disease activity. We continued those weekly follow-up reports, ended up being about 22 until July 2nd, when we notified the OIE that we were considering the disease stable or endemic in the 7 affected southwestern states, as well as Washington state, because of the detection of disease in wild and or feral rabbits in all of those states. This has created a little bit of confusion, so just to clarify a bit more, RHDV is still a reportable disease in all 50 of our states, and data collection is continuing on investigations that occur around RHDV. In the stable states, new information, positive information will go into our next 6-month report to OIE, while a finding in a new state or a new species will be immediately reportable. Next slide, please, Julie. So in addition to allowing us to change our reporting time frames, this change in status also allowed us to implement some alternative testing approaches. The status of RHDV as a foreign animal disease initially limited all of our testing to NVSL fattle. And we rapidly discovered that providing support for the larger scale testing, particularly in wildlife, that testing being desired by the agencies managing the disease in wild populations, was going to be a challenge for us. As Julie mentioned earlier, we don't have regulatory authority or program or funding around RHDV. So we didn't have that funding to support a large amount of testing, and also COVID was impacting our laboratories. So with the shift to the stable status, we were able to provide, actually to support the expansion of laboratory testing in the affected stable states. And in this approach, SAHOs in the affected states were allowed to designate a lab in their state to provide RHDV2 testing for domestic rabbits, wild lagomorphs, or for both. The limitation of this was that these state-designated labs were limited to testing submissions originating from their own state only. But the added flexibility was that these investigations in those states don't have to be conducted as a foreign animal disease investigation with an FADD being involved. In addition to National Wildlife Laboratories, the National Wildlife Health Center and the Southeastern Cooperative Wildlife Disease Study were approved to provide testing for wildlife submissions from any state. So they were came online to do national level testing for wildlife agencies that needed testing for their animals. FADL is continuing to provide testing support for domestic rabbit samples from all of the RHDV2 foreign animal disease investigations in states outside of those stable states. They're also continuing to provide support for domestic rabbit investigations in states in those stable areas as needed. So if a designated lab wasn't identified or if there's some other circumstance causing difficulty, the samples can still go to FADL. The samples just need to be submitted as a foreign animal disease investigation. And finally, while RHDV2 may be here to stay with the wild and feral rabbit involvement, RHDVA is not present in the U.S. So we definitely need to continue to monitor for this disease. Those of you that are part of the laboratory expansion plan have seen that we have an instruction that if samples go to your lab and you rule out RHDV2, but you suspect that RHDVA could still be involved, samples should then go on to NVSL so we can ensure that we're still monitoring for RHDVA as well. So that's all I have now. Back to Julie.
Thanks, Lynn. Appreciate it. And I did see a question pop up in the chat that I'll answer now, and then we can do the rest of the Q&A after. A couple of folks have asked for the difference between feral and wild rabbits. When we talk about our big categories, our domestic rabbits are European lineage domestic rabbits. So those are rabbits in confinement, either because they're a pet and they're in our backyard or in a hutch, or they're raised for domestic purposes. A feral rabbit is a European domestic rabbit that has escaped confinement. Either the people set their bunny free and it moved on to different areas, or it escaped its hutch or left the yard. Rabbits are a bit opportunistic, and they're fairly good at fitting into new environments, and rabbits also procreate like we think that they would. So if a couple feral rabbits find each other, they can mate and make little rabbit babies and form a little feral rabbit colony. But those are all domestic rabbit origin, where wild rabbits are the species that we talked about earlier and that Tom gave the summary. Those are the cottontails, hares or jackrabbits, pika. So those are in the wild category. So different genus and species between wild and feral. Feral always have a domestic origin as far as their lineage. Apologies, we didn't explain that at the beginning.
Julie?
So I'm just going to, yes, go ahead.
Before you go on, just another quick correction, thanks to Andrea Michelon. I left out Zacatecas in the Mexican states that are involved, and also San Luis Potosi, which I didn't think I had in my list. So thank you, Andrea, and just that brief correction. So 9 Mexican states involved.
Okay. It's still within the northern half of the country, more or less?
Yes.
Mm-hmm. Okay. Thank you, Andrea and Lynn. Okay, so I'm just going to give a quick summary of outreach and available resources, and I believe that the link maybe won't work, so I added it to the chat box to get you to the STIA homepage, the Center for Epidemiology and Animal Health. On their risk identification are all of our resources for RHD, so you all have that link also in the chat box. So because of a gap in knowledge and kind of a rapidly evolving situation, again, since March or April of 2020, we've worked with our partners at STIA and multiple groups to create some information. So there is a disease summary sheet and a rabbit disease fact sheet. We worked to create a cleaning and disinfection guidance. Again, the environmental hardiness of this calicivirus is particularly noteworthy. That information was also translated into Spanish. Earlier this year, we were able to work with the Environmental Protection Agency to ask for a list of disinfectants that should be efficacious against RHD, and that's called List O, which we've linked on the page. And then, as Lynn went into, there's some nuances with the vaccine importation and special permits. So we created a frequently asked questions document with our colleagues at the Center for Veterinary Biologics. So that information is all on one page, easy access. And then that box on the bottom that says interactive map, that takes you to a county level representation. That is the information that Lynn went over with the blue boxes on the county level. And again, that's being updated periodically if there's any new detections or further geographic spread. We've also worked pretty hard to bring in all of our partners and knowing that we have lack of regulatory authority and that there's other folks with better boots on the ground. And again, the time frame for this is relatively uncanny. The entire time we've been working on this outbreak, we've also been under travel restrictions due to COVID. So we've had to be pretty creative to, to pull all that information together because, field examinations or for us going out in the field really haven't been possible for the last 6 months. So we've relied heavily on our State's Department of Agriculture, and wildlife, and again, that's since 2020. We're also including New York because of the isolated incidents in the Manhattan area, and then our 7 states. The labs have really stepped up, both Plum Island, as Lynn talked about, and then our state-designated laboratories and the 2 national wildlife labs have really helped us expand capacity, and then outreach involving all those stakeholder groups. So we have the meat industry, ARBA, the American Rabbit Breeders Association. We've worked with APHIS Animal Care because they do a lot of the inspections in some of these breeding facilities, and then zoos, other wildlife partners. The House Rabbit Society is a big social media group for pet owners, and fairs and exhibits, which the rabbit shows more or less have been canceled or severely restricted, again, due to COVID, but we've really worked with that group to make sure that any appropriate biosecurity and quarantine type of activities have been put into place to avoid further spread of the disease. Thankfully, we've gotten a boost from some popular media, and again, going back to the New York cases, this story was picked up by both the New York Times and the New Yorker magazine. Those are the two outside captions, and both fairly good scientific articles, I would say. And then very recently in JAVMA, the disease was summarized, and that was just about a week or 2 weeks ago that story came out. So several national level reports have picked up the story, so I think it certainly helped reach veterinarians and other interested groups. And this is just my shout-out page. So again, kind of circling back to that beginning coordinating cell, again, with the 3 business groups of veterinary services and wildlife services working together. But we really realized that while VS is sort of the lead and has the responsibility for the OIE report, it has taken combined coordination with Canada, the Canadian Food Inspection Agency, and Seneca in Mexico, and then again our state agencies, both agriculture and wildlife. Veterinarians are a big bucket as well. We've got our industry and commodity veterinarians, our private practice, sort of the exotic vets who take care of rabbits, our foreign animal disease diagnosticians who've done a lot of these initial work, the labs, National Wildlife Health Center and SCWDS, both helping on the wildlife side, and our state-designated labs, and then our own colleagues at Plum Island who were responsible for the initial crush of samples that came in, helping with the phylogenetics and that whole genome sequence, that part of the epi-analysis, and also getting the lab expansion going, so providing those standard operating, those SOPs to get the labs pulled on, and then our industry and stakeholder partners. So a quite combined effort. I also want to make sure we mention our colleagues at other folks at the Department of Interior. National Park Service has had some case detections, and our Bureau of Indian Affairs and our tribal partners. A lot of the land in the Southwest is adjacent to or involves tribal lands, and we've wanted to include our tribal partners there as well. So this is just a snapshot of why we're working on this. We do have this diverse group of what we all call rabbits, even though some of them are hares and jackrabbits and cottontails. So again, both our fancy show rabbits all the way over to our picas down there on the lower left. And closing here, all 3 of our emails are available. You can reach out to any or all of us for follow-up. And I'll stop there, Liz, and hand it back to you for question and answer. Thank you.
Yeah, we have a lot of questions in the chat. How do rabbit rescuers determine disposition of feral rabbits? Wild rabbits, domestic rabbits, is this an issue in this current spread?
I can take that one, Julie. This is Lynn. So as Tom had mentioned, because of the environmental persistence and hardiness of this agent and the fact that there's a tremendous viral load in affected rabbits, we're definitely concerned with how carcasses are handled and do believe that it could contribute to spread. So I think your best bet is to always go to your state animal health official to find out what the best options are for you in your particular situation in your area for disposal.
Okay. The next question is, when is the key epi info going to be released? We recently had a domestic rabbit die and decided not to do a FAD as there were no risk factors for exposure to any other rabbits. Interested in the vector hypotheses.
I can take the first stab at that. So our EPI team includes the epidemiologists in those states, and Brad Christensen has also been involved and is working on that part of it. So we have the two components of that. Further phylogenetic analysis coming from the kind of a second wave of these submissions, and these questionnaires. We have epi questionnaires from positive detections in those states, and those are mostly in the domestic side. So as we get to evaluation of that, probably in the next month or two, and again, we're working on with our epi team and with FIA to get analysis of those questionnaires. We'd like to hope by the end of the year for sure to get that analysis done. And as Lynn mentioned, we did get the GenBank sequence from Mexico, which may also give us a clue as to potential which side of the border did the introduction come from, or are we clonal and we truly can't tell if it's all the same virus. So further analysis is coming. But my suggestion is if you have signs consistent with RHDV, A or 2, submit it. Really, I think that the more that we know, then the more that we learn. And we've certainly had negative submissions from the surrounding states, but I think it's fair to look under every rock at this point. So if you do have signs that are consistent, again, as Tom pointed out, that sudden death, hemorrhagic disease, whether there's any risk factors or not, it might be worth submitting just to get an idea, or for peace of mind to say that we've got a negative there.
Okay. Is there a company that is working on approval in the U.S.?
Tom, you want that one?
Sure. We're talking about vaccine approval?
Yeah.
Is that right?
Vaccine, I think.
Okay. Yes, there's at least one vaccination company that is considering pursuing the vaccine in this country, but as of yet, to the best of my knowledge, they really have not had much progress yet. Unfortunately, there's not, no matter what, there won't be very much money in such a product. So I think that's always going to be a problem when it comes to folks putting the investment into the vaccine that's necessary because it's such a small industry. But we do have one company that has been talking about it, but so far little progress has been made.
Can any of these cross-pollinate?
The viruses or the species? I guess let's go back to that that submitter and give us a little bit more information.
Okay. So moving on, what is the status in Canada?
Oh, I can try that one, Julie. And I would invite, if we have any Washington or Canadian colleagues on the line, to weigh in on chat as well. But my understanding is that at this point, the disease is still limited to BC, and I think their findings were strictly in feral domestic rabbits.
Yeah, and that was the next question, outside of BC.
Again, to my knowledge, I don't think it's outside of BC.
And for the question, can any of these cross-pollinate, it was meant as a species, a wild and a feral.
Oh, can a feral rabbit mate with a wild species rabbit?
Right.
To the best of my knowledge,
no, right?
Yes, correct. To the best of my knowledge, hybrids don't exist. Our expert, Jeff Root, would be the person who could answer that definitively, but it's my impression that they do not hybridize, they do not produce.
The next question is, was the origin of the New York City case identified?
All of the cases in New York, and I don't know if Todd or Leslie are on from Veterinary Services, but all of the cases, and I believe there were 13 total detections in New York, were all centered around one veterinary clinic. Either the rabbits had boarded at that clinic or had been seen at that clinic. The first case was introduced from a rabbit rescue. So the case 0, I guess, was a rescue rabbit that had entered New York and died shortly after, and then the sample was sent to Cornell. the actual origin of where that rescue rabbit got it is undetermined, but the introduction into New York was from a rabbit that came in from Brooklyn, again from animal shelter from the rescue circuit.
The next question is, have you seen any decrease in cases since using the imported emergency vaccine, or is it too soon to tell?
Well, certainly in the curve of the outbreak on the domestic side in the 7 states, we definitely seem to be on the downward side. Not as many reports of new investigations. Whether that's due to the vaccine or not, that's hard to tease out at this point.
Okay. Has rabies been included in the differential list for acute domestic rabbit mortality, especially for those housed in indoor cages? I cannot recall any differentials on your slides.
This is Tom. Rabies always has to be included in the differential. Not very frequently encountered in lagomorphs, to my experience, but one always has to be aware of it and keep that in mind when it comes to a PPE. So yes, it definitely needs to be on the agenda.
Okay. Will herd immunity take hold in the Southwest and slow the spread of the virus, or will this virus not work like the COVID virus we keep hearing about?
I could give that one a go.
Go ahead.
And Julie, you can fix what I mess up, maybe. Certainly, and Tom had mentioned this as he was discussing the call we had with wildlife colleagues last week. Certainly, we hope that herd immunity at some point will help, especially on the wildlife side, but it's hard to tell at this point how that's all going to look.
Okay. Do we know how go ahead, if someone else wanted to
Yeah, this is Tom. The attack rate in our experimental work was about 60%, and we're going to repeat that work again to get a better handle on it, but it's our impression that the attack rate in our wild lagomorphs is less than the attack rate in our domestic rabbits. That's just a subjective gut impression. Thank you.
Do we know how the California virus is genetically related to the Arizona and Mexico virus detections?
So far, and our team at Plum Island did an initial sequence comparison, and I believe that was Colorado, California, Arizona. We have not yet compared to Mexico, but during the first round of phylogenetics, the virus in both domestics and in wild species was clonal, identical virus in those states, but distinct from what we had seen in New York and Washington. So the Southwest were clustered together, different than the northern detections, and again, the same virus in both the wild species and the domestic species. We will be repeating some of those phylogenetics or adding on more samples as cases have come in, again, in our feral populations in Colorado and Utah, and now that we have the genetic sequence from Mexico. So stay tuned, because a second round of whole genome sequence and genetic analysis will be coming as our Plum Island folks have time to do that.
Thank you, Julie. Just a small correction, though. So the comparison we have right now is Arizona, Colorado, New Mexico, and Texas. So California, Nevada, and Utah, they're still working on sort of getting all that information in. So anyway, more to come later, hopefully, on those.
So do we know where the rescue rabbit entered New York from?
I don't know that we know the origin. I know that it was within a rescue circuit and then came in into Brooklyn into an animal shelter was in kind of the rescue foster network, and then got to the veterinary clinic. The previous origin, I'm not sure that we got that traceback information.
Okay. And do any states have any rules about moving rabbits into their state?
That's a great question. So there's a very mixed regulatory authority. Some states have authority over rabbits, and some states have no regulatory authority at all over rabbits. They're not even listed as an agricultural species. Both California and Washington State have put in some movement restrictions, but interstate movement of rabbits is not a controlled activity.
Okay. You have mentioned this earlier, but what are the estimates of mortality rate in wild rabbit populations?
Tom, do you want that one?
Yes, you bet. As Lynn said, we had a call last week with all of the state wildlife agencies involved to try to get somewhat of a handle on that. And truthfully, all of the information is really, really circumstantial and subjective, and our general overall impression from that call was that it's not as bad as it could be, and in places where the disease has gone through, they're still seeing young rabbits as well. So I guess the answer to your question, we don't have the answer to that, but just from dealing with the folks in the field, it sounds like it hasn't necessarily decimated that or completely annihilated those rabbits, even though it has pushed them back.
Thanks, Tom. Yeah.
Answer, but we'll have. But I think as time goes by, we'll have a better feel for what exactly happened. Thank you.
Thanks. Yeah, and overall mortality rate, kind of a general guess, is between 50 and 90% in domestics, and we have had some premise report 100% mortality loss. It just wipes through and takes them all out. So again, subjectively, it looks like the wild populations maybe have had a little bit more ability to withstand that, and then again potentially have some immunity and repopulate.
The next question is: it's my understanding that in visiting the MedGene labs, is that they are ready to do clinical trials on a vaccine, yet lacking funding to continue forward at this time.
That is our understanding as well.
Okay. Cottontails and European rabbits do not produce viable offspring. However, there are some documented cases of hybridization with hares. I think that's just a comment.
Excellent. No, that's good to know. So potentially cross-pollination with hares. I assume that's one of our wildlife colleagues chiming in.
Thank you. Thank you very much.
Okay. Do rescue groups ship rabbits around the country as they do with dogs and cats? They bring rabbits from other countries that have been rescued from meat markets like dogs.
I don't have knowledge of international importation, but certainly domestic movement and even North American movement. At one point pretty early on, I think it was probably April or May, we had information forwarded to us from one of our spies on the ground, and they said this rabbit rescue is planning this trip starting either in Saskatchewan or Alberta, and they had mapped out their road trip to go to animal shelters and rescue and working to kind of pull animals out of hoarding situations. And the trip went from Montana, Wyoming, Colorado, Arizona, New Mexico, California, up the Pacific coast, and then back. And there were people's heads were exploding. Number one, because we were under COVID restrictions, of course, and asked not to move around. And also because we knew we already had detections in those lower states, specifically Arizona and New Mexico. So going into that state, pulling rabbits out and then moving them to an area where disease was not detected was certainly worrisome. So yes, very networked amongst the rescue circuit, and pulling animals out for various reasons. Again, animal shelters, humane societies, and SPCA agencies, but again, and then also hoarding and those type of situations, and they'll move them into foster and rescue circuits.
As of now, the virus only affects rabbits. Does this virus have the potential to change and later affect other animals such as raptors or predatory species?
Right. I guess if you interpret that literally, I guess it always has the potential. However, right now it appears that raptors and predators can spread the virus. The virus can pass through them and remain infectious, but so far there has been no evidence that the current either one or two causes disease in raptors or predators. We saw an interesting thing early in this outbreak in that dogs were appearing to be ill, and the question was whether these dogs had just eaten too many rabbits or whether there was actually something going on, but there is no documentation of the disease in carnivores to the best of my knowledge.
This is Lynn. Someone, I think, in the chat gets at this a little bit further down the line, but probably the bigger concern would be the impacts to predators and raptors by the disease impacting their rabbit prey base.
That's a very good point, Lynn. Very good point.
Oh, I stole it from someone else who chatted it.
How are Southwest and Mexico outbreaks chronologically related?
So this is Lynn again. I need to look back and see, and perhaps in our next call with Mexican colleagues, I'd really like for this question to be asked when their first cases actually were. But my remembrance of that is that it was just pretty much concurrent. When we started finding or identifying the disease in New Mexico and Arizona, we were hearing that the same kind of mortality was going on right across the border. I would invite any of our state colleagues that are on the line that might have more specific information to go ahead and chat that in too so we can see. Julie, you're nodding. Do you remember anything different? You've been on the Mexico calls too.
Yeah, I would say exactly that, pretty chronologically matched. So our detections in the Southwest popped up middle to end of March, but we did have especially wild rabbit mortality events backing up to early March, February, and the same for our Mexican colleagues.
Another question is, the larger question is, what is the consequence of wild rabbit mortality to their predator species?
Tom, you want that one? Yes. In Europe, we have seen declines associated with the removal of their species of interest. So it's possible. It's definitely possible, especially probably not so much our common predators and raptors, but the varieties that are more threatened. So it's surely something that we worry about. But one of the things that we like to fall back on is that rabbits have been, or lagomorphs have been particularly resourceful in the past. Again, we mentioned Australia as being a good example. So while it is possible, and while threatened and endangered species would be particularly at risk, hopefully that will not come to pass. But they have seen some of that in Europe, and it surely is possible.
We do have a link in the chat for everyone from Luis Lopez, which is their RHD link, if anybody wants more information. Another comment is, we know that there are rescue groups that have come from Canada to pick up rescue rabbits from the U.S. to take back to Canada, as well as U.S. rescue groups who have gone up to Canada and brought rescue rabbits from Canada to the U.S. And then the next question is, if a collection of domestic rabbits, meat or show rabbits, tests positive, will the herd be put down if all don't die on their own? I've seen an Excel spreadsheet showing cases, and there were examples where not all rabbits that they were still alive were euthanized.
This is Lynn, do you want that one, or you want me to give it a go?
I'll give it a go, and then you guys can chime in too. So it is decision between that producer and their state animal health official. And I can tell you that the response has evolved. In the beginning, when the detections first happened, and because of some of the regulations in states involved, depopulation was a response, and that over time, as the disease was found in wild rabbits, some of that response has changed to the choice of long quarantines. So it just depends. And Julie, anything else to add that I sort of missed on that?
No, I think that's accurate. Mexico, as we've been working with the Senefica team and our folks in international services, Mexico has taken a much more drastic step. They essentially do stamping out, depopulate any of those. And Mexico's a little bit different industry where a lot more rabbits are raised for meat. But they have done full stamping out, and in some cases switch the production to a different species. So a rabbit farm will then turn into a poultry farm after cleaning and disinfection. We're also following up with some of our domestic detections on cleaning and disinfection, fallow period, and then restocking. So again, a little bit more information will be coming as time goes on, and we can get a better handle on the state-by-state responses and what folks have done.
Good additions, Julie.
Liz, are you still there with us?
Yeah, do we have any other questions that might want to come through the chat?
Okay, ladies and gentlemen, just to reiterate, please be sure to select all panelists from the drop-down menu in WebEx chat before you type in your question and press enter on your keyboard. Thank you.
I don't see any others. Go ahead.
Okay, thanks. So I think Louise has put some information in as far as links, and I did as well. Again, in the U.S., certainly many states have posted RHD resources from the State Department of Agriculture. I should also point out that our colleagues at the National Wildlife Health Center are reporting and tracking the wild mortality events on their Whispers platform. So that's an additional spot for information. And then again, our risk identification page within SIA has all those resources in an interactive map that's updated by our GIS team. So lots of resources out there. Double check based on credibility. We have had some misinformation and some rumors, so make sure you're checking in with a credible source. But certainly the state and national agencies are pretty solid options.
Well, that's great. If we have no other questions, I'd also like to thank all of our speakers for today. And thanks for everybody who joined. We do have another webinar tomorrow at 2 o'clock Eastern Standard Time. And the topic is evaluation of above-ground burial decomposition in a simulated African swine fever disease outbreak. And the speaker is Dr. Keith Bailey, Clinical Associate Professor and Comparative Pathologist at the University of Illinois College of Veterinary Medicine. And if there's no other questions, I'm going to bid everyone to have a great rest of your day. And thanks, everyone.
Ladies and gentlemen, that concludes our conference. Thank you for using AT&T Event Services. You may now disconnect.